Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Jackson Mace, a neuroscience PhD candidate at Johns Hopkins University. Jackson is also a 2023 Toffler Scholar. Today, we will be discussing Jackson's efforts in understanding the mechanisms of neurodegeneration occurring in MS. Welcome, Jackson.
Thank you. Happy to be here.
So let's just kind of jump in here. What influenced your decision to pursue neuroscience? Because that's a pretty hefty career.
Yeah, I think, to be honest, it was a little bit of serendipity mixed with previous experiences. So kind of like a clashing of different ideas and dreams and aspirations and so forth. But I think the driving factor behind a lot of it was really this ambition to take part in scientific research, science, explore the unknown, whether that be space or the human body. It just so happened to be the human body through different experiences as I progressed in science. And specifically neuroscience, I think I was mostly inspired by my dad, who I had seen as I grew up. He had been living with multiple sclerosis. So that's a neurodegenerative disease of the brain. And I knew about the brain, and I thought it was really interesting. The brain, controls behavior. It controls appetite, but it can also go awry and you can have neurological diseases. So I thought there was a lot to uncover and understand by learning about the brain.
How old were you when you were, when your dad was diagnosed and you were going through that with him?
Yeah, so he was diagnosed, I believe, in his mid-30s. So that's when I was born. So basically my whole life. And I had never really noticed it until later on, as I got older and my parents had more of a discussion about it. My dad was more open to talking about it and some of the struggles and so forth. So I think really around middle school, early high school is when I really started to become more interested in what was going on. And I paid more attention to, minor differences, even in a few years span of how he potentially was affected by this disease and how it affected his walking and such. And that really, while it was, it's hard to see if the disease does progress, it's really interesting to consider something in the brain that can control something, like I said, like movement and such.
Yeah. It sounds like they were pretty open, or your dad was pretty open to have those discussions with you too, even at a young age.
Yeah, definitely. And luckily, like my dad's still around, and he's the disease luckily hasn't progressed so much in the past 10 years, which is great. And potentially that's due to the innovative therapies that have been, coming out over the past decade. But I think he is also very much a free thinker, scientist type of person. He's really interested in everything from biomedical engineering to drugs that come out for diseases. So I think he also really sparked my interest in discussing these therapies for diseases like his, and much, much beyond just multiple sclerosis, things like Alzheimer's disease and such.
That's fascinating. So you did come from a family of scientists and engineers, and so that was in the house even at a young age.
Yeah, I think so. And those paths, I have three other siblings. They've taken different paths. Some of them have come back to STEM, some of them are in STEM, but I think we're pretty well spread across different fields.
So talk about that journey through college and undergraduate and now, your PhD. What was kind of that journey?
Yeah. So like I said, it was kind of, I kind of went out on a whim with choosing neuroscience. I thought that the brain was really interesting, and that was mostly the driving factor as to why I chose neuroscience, check that box as my major for a lot of schools. I was pretty confident that I wanted to do science, but didn't exactly know it was neuroscience. So moving into college, I had immediately as I got to school, I took a neuroscience introductory course with a professor named Dr. Eric Roth. And this entire class was really built upon the foundation that zombies have this manifestation of different clinical disease, essentially disease facets. They have different gait, they have different movement styles, they have different tonal vocal aspects in how they speak. So there's a lot of things going on in zombie. And I thought this was really interesting because it kind of set the whole atmosphere to how much the brain does control. So like I said, whether that's drive or how you talk or how you move. And that was really fascinating. And of course, the class was done really well for these introductory neuroscience students. So immediately I was really hyped. I was excited. I thought this was probably the major that I wanted to stay in. And this kind of took me towards looking at research. I knew I was interested in diseases. I wasn't positive that that's what I wanted to do. I thought everything in the brain was interesting, but I stumbled upon a lab. Another, lucky moment. I took the opportunities that I had. And one professor communicated with me a lot, Dr. Amy Griffin, who ran a spatial working memory lab. So a lot of memory aspects in rats. And she was great. She communicated with, a confused and not very experienced freshman in college. So I went into that lab about halfway through my first year. And again, it really sparked my interest for science. This was beyond anything I had ever done. I had always, read little passages, maybe for literature courses and such in high school or different scientific articles. But now I actually felt like I was kind of creating these scientific articles. My role was very minor as an undergraduate student, but I still felt really excited to be in that world where I was stimulating the brain of these rats to see how it affected their memory. And that's what really took it off. So a mixture of the professors that I met, the classes that I had, but really the laboratory experience throughout college.
Yeah. What an innovative way to teach a bunch of students their first neuro class is to use a zombie kind of analogy there. That I bet you that was fun.
Yeah. And the movies that we were able to watch with it and dissect. And then there's a book that went along. It was great.
Yeah, that's awesome. People can't see you since we're only recording audio, but you're actually sitting in a lab right now. Some of the little beeps and squeaks back there, and I can see all the little kind of test tubes and everything in the back. Talk about, what that means, that first kind of lab experience that you had with Amy Griffin, Dr. Amy Griffin. What did it feel like being in the lab? Because that's a special place.
Yeah, definitely. I think something that it provided for me was a place of purpose outside of just working in my courses. It provided me with a place to go outside of being in my home or the library. So there is this really cool, to me, a freshman in college, a really cool laboratory that I could go to. I had the key and everything. I could walk in, go to my little designated area, and I could hook up these cords to the top of a live rat, and basically monitor how they are thinking to an extent when they're making decisions. And that aspect of my life was really unique from my peers and my friends. And I love to talk about it with them, but I also love to engage with it. And I think having that opportunity to do something very unique and very foreign to me, but also really, really exciting, was something that I wanted to continue to do and strive for.
So you graduated undergraduate, and then you went to your graduate program. Talk about that.
Yeah. So one thing that I didn't talk a lot about was the summer experiences that I had done that also really contributed to my, not end goal, because I'm very much in the beginning, but where I'm at now in my PhD. So I had done, like I said, I had done a lot of undergraduate research with memory in rats, but I didn't really get to see the medical side of things in neuroscience or really in biomedical sciences at all, because my university didn't really offer a lot of biomedical research. So in my summer internships, I had the opportunity to take part in hospital research, or, I worked at Washington University in St. Louis, which is right at the medical campus. So another really biomedical world. And from there, both of those experiences really opened up the doors to medical research, specifically neurodegenerative disease research. I was fortunate enough to have the opportunity to do some of that earlier on before I started grad school. And that really confirmed altogether that neuroscience, medical neuroscience, was what I wanted to pursue. So that's where I landed now at Johns Hopkins, doing really biomedical research in the neuroscience realm.
And what was the hospital that you worked at in that summer program?
Yeah, so that was Nemours Hospital in Wilmington, Delaware. So I went to the University of Delaware. So this was about 30 minutes away, or 2-hour bus rides away, but it was worth it.
Did you have exposure to adults, or was there, because I know they have a children's hospital there too?
Yeah, so it was actually all children's research, the internship. And this was the first, similar to when I first started in college as an undergrad, this was my first exposure to something that I had really spent a lot of time applying for and had to get chosen for. And then it was a totally new environment for me. I had never worked in a hospital. So I took what was offered, but also that experience was really good. They have a well-established summer program. So all of the research I did there was actually pediatric research.
So did you interface with the children patients?
Yeah, we did. They had like I said, they had a good program that really exposed these mostly undergraduate researchers to the world of medical research. So I had met two really, young cancer patient children. I was doing mostly cancer research there, so not so much neuroscience, really just kind of dipping my feet in the medical realm of research. And I also had the opportunity to witness my boss, the principal investigator of the lab. I had seen different patients come to the lab and talk with her and discuss what's going on and how much the scientists and then the patients really connected. And that was really special to me and something that I didn't really see in my college experiences so much.
And I think around this time too, wasn't there like an Amgen scholarship program, or was that a part of funding this internship too?
Yeah, so the Amgen Scholar program was the next one at Washington University. And that was the next summer where I took what I liked. So I knew I liked neuroscience. And then from this cancer research internship in the hospital, I knew I liked medical research. So the Amgen Scholar program, again, it's for undergrads that are pursuing research, so likely a doctoral degree later on. And I studied medical neuroscience research or neurodegenerative disease research for the most part.
Yeah, and you talked about the kind of witnessing where the clinician and the scientist, kind of this interaction or this convergence, if you would. What were, was that kind of the big takeaway, or what were some of the big takeaways or experiences that you walked away? Because that had to be a pretty, to me, it would be pretty powerful, but it also probably be a pretty emotional kind of time frame too.
Yeah, definitely. So I think the pediatric hospital research was kind of a shock to me, to see these patients in real time and to witness these interactions between, my lab and the director of the lab and the young pediatric patients and their parents. But then the Amgen Scholar Research program, which was the next year, where I did neurodegenerative disease research, that was a whole different playing field, because at this point I had become much more well-versed in the field. And I knew a decent amount about neuroscience, and I connected with, again, the principal investigator, or the boss, my boss of the laboratory. And he was most of the students are paired with MD, PhD, clinician scientists, physician scientists, and such. So we had the opportunity to go on grand rounds and see the disease that we're studying. So I had been studying mostly Parkinson's disease, but as well as a little bit of Alzheimer's disease. So I had been able to see a lot of the movement disorder related and then dementia related patients. And this was really, really interesting for me because I was pretty confident at this point that this is the research I wanted to do. So to see the exact type of patients that I was working in the lab to hopefully make a positive influence on with regards to therapies, that was really powerful. And of course, it made me think back to my dad with his disease and really giving me a lot of this internal drive to continue what I'm doing, because there's a whole spectrum of patients, I think. Like you said, this was, while it was a good experience education-wise and research-wise, it really provided and opened up that spark, that flame for myself in regards to pushing forward and wanting to pursue the research that can hopefully help these patients. Because like I was trying to kind of discuss earlier, that there's a spectrum of these patients depending on how sick they are and how affected they are by those diseases, and whether therapies are working for some patients or not working for other patients. And I think that is a lot of those differences and disparities between the patients and how well they, respond to some of the therapies was really also a motivating factor, because you want all the patients to get better and you want all the patients to respond to the best possible therapies. So that was really powerful to see, I think.
Yeah, that's amazing. It's amazing. And you talked about that spark, and I think you just mentioned just in that those few minutes there, the push to keep moving on and doing the research, because it's how long have you been going to school? It's been a while, right?
Yeah, grade like 20 at least.
20 years, and you're right there as a candidate for PhD. Have you defended yet your?
So I'm very close, and I'm supposed to defend in a month and a half, something like that.
Wonderful. Well, I know that that will go well. So talk about what this current research is that you're doing, and specifically around trying to understand. You mentioned these therapies that sometimes work and they don't work, and I'd imagine it's dependent on the way the body and cell death or mutation happens. Can you describe your research?
Yeah, absolutely. I need to limit myself here so I don't ramble too much. 3-minute elevator pitches. So I'm studying multiple sclerosis. Again, similar to a lot of my previous stories and such, it kind of happened through luck and serendipity that I ended up studying the disease that my dad actually has. So that has been a really driving force for me as well, because, my father's living with this disease. So of course I want to provide the best possible research and therapies for that disease. And essentially I'm studying the pathology that occurs in the brain and the spinal cord, as well as the eyes. So really that whole central nervous system of MS patients. And I use mouse models mostly to study this. So we use a EAE, or experimental autoimmune encephalomyelitis mouse model. And this mouse model really recapitulates the disease facets of multiple sclerosis, which is characterized by immune cells that infiltrate and move into the spinal cord and the brain, and they essentially attack the endogenous tissue that's there. And that obviously causes a lot of issues related to movement and thought and so forth. So that's what I'm studying now. And a lot of the times MS or multiple sclerosis is considered an immune disease or an autoimmune disease or a neuroinflammatory disease, but it's really a heavily, it's becoming more known as a neurodegenerative disease as well. So there's very much a loss of the gray matter, the neurons that help orchestrate, like I said, things like movement and such. So I'm studying specifically the neurodegeneration that occurs in MS patients. So looking at these gray matter lesions or neuronal lesions that occur in the spinal cord and the brain. And I'm studying really the cell death process that occurs as a downstream effect of many of the other things that are occurring at the time, being where an MS patient may be going through something like a acute attack, or potentially they don't have an acute attack going on and they're older patients, but we still see this chronic neurodegenerative phenotype occurring in these patients.
You mentioned eyes, and what about the eyes?
Yeah, so MS is a really interesting disease in that it affects a very wide span of tissues. So a lot of neurodegenerative diseases like ALS and Parkinson's specifically, and a little bit of Alzheimer's are really focal. They affect a certain susceptible type of neuron in the brain or something like that, or in the spinal cord. But MS, you have these immune cells orchestrating a wide phenomenon of overactivity throughout the entire central nervous system. So that embodies the spinal cord, the brain, but also the eyes. So, and a lot of the nerves around the eyes. So the optic nerve, which connects basically the eyes, the retina, to the brain. There's immune cells that attack those areas. And in MS patients, as well as our mouse model, we see a very prevailing loss of the retina. So the retina actually gets thinner and thinner over time. And this is because there's neurons in the eyes. They're called retinal ganglion cells, or RGCs. And these cells actually die over time.
So what is it about the cell death that we know right now? If you have MS and now it's doing something to that gray matter, you said, what do we understand today?
Yeah. So that's a great question. And we, like a lot of science, we don't really know why these cells are dying and what the overarching mechanism of cell death is. So there's quite a few different cell death pathways that the research fields have already come up with in many different diseases. So there's the typical apoptosis, which is a very regulated form of cell death that's occurs after there's DNA damage. And this is studied in cancer fields and neuroscience fields. So there's a lot of cell death pathways that have been described. But one cell death pathway that is newly described over the past 10 years really is a cell death pathway known as Parthanatos or Parthanatos, depending on how you want to call it. And this cell death pathway was really coined by the Dawson lab at Johns Hopkins. So they have been studying Parthanatos in Parkinson's disease for the past decade. And from there, it's really budded out across the world. So other labs have studied it. They've shown this cell death to occur in diseases that it was not known to occur in before. It's really interesting. It has its very unique aspects that make it specific to that cell death pathway. And this is important because all these cell death pathways, while there's a lot of cell death going on, there's key players that execute the cell death pathway. So there's certain protein biomarkers, like you hear in the pharmaceutical companies and such, that can be targeted. So we need to know what types of cell death are occurring and when they're occurring in order to target the correct proteins to stop them from occurring.
Do we know what those proteins are yet, or is that still being researched?
Yeah, so for most of the cell death pathways, including the one that I study, mostly Parthanatos, we do know a lot of the proteins and the exact way that the pathway is orchestrated and how it's carried out. But what we don't know is often how it starts. And we also don't often recognize the key switches to where it may switch from one cell death pathway to another. And this is really where my research comes in.
So what's the overlap with other parts of or other systems? Is there a connection to, the immune system or the cardiovascular, or is there, you started with MS. We believe that MS is an autoimmune. What do you think are the other connections here inside the body?
Yeah, that's another great question. So that could be answered in two parts. So one aspect of it is that a lot of the insults that cause these cell death pathways to ensue, so something like maybe in the brain and in the heart, a lot of these cell death pathways occur in different organ systems and in different tissue systems because the triggers for those cell death pathways are similar. So that's a lot of my research as well. A lot of the immune activity we think that is causing the cells to die, specifically the neurons, which are really important cells to preserve because they can't really, we don't know of ways that they can rejuvenate and repopulate. So when they're gone, they're gone. A lot of these triggers that cause these cells to die are things like DNA damage or oxidative stress, these kind of catchphrases, but they're really important because you can have, for instance, a lot of these reactive oxygen species or oxidative stress occur in something like ischemic stroke, but you can also have that occur when you have an occlusion in the heart and you have issues with providing the proper oxygen or things like that in the heart. And then you can also have an immune disease. So you can have something like a joint-related immune disease, something even like arthritis or GI tract inflammation. And these immune cells can actually release these reactive oxygen species that cause DNA damage on cells in those local areas. And those cells can then degenerate and die over time, whether they're epithelial or skin-like cells, or whether they're neurons or cardiomyocytes. Those cells are undergoing very similar insults and they can die in a similar fashion.
We hear a lot in the news about inflammation and, all of that, especially, when it comes to maybe dementias or Alzheimer's. Is there a inflammation connection that may be occurring?
Yeah. So you nailed it. Inflammation is becoming a really hot topic, especially in the field of neuroscience. So the field of neuroimmunology in general is just, in my opinion, of course, I'm very biased, but it seems like very much the fastest growing field in terms of neurological disease research, because so many of these diseases, while they have very unique components that are cell autonomous, so they're specific to cells and neurons dying, there's often, I would argue, always an immune component, whether that means the immune cells come first, like in the case with multiple sclerosis, or whether the immune cells eventually come around, like in the case of many neurodegenerative diseases like ALS and Alzheimer's and Parkinson's, where there's this clash of the disease phenotype ongoing with a mixture of immune cells that are also kind of cleaning up or trying to clean up that cell death, really that whole scene of cell death. And that causes that neuroinflammatory cascade to ensue and cause more damage oftentimes, which is what we're trying to prevent in multiple sclerosis.
So you mentioned biomarkers, you mentioned the pharmaceutical companies trying to, are you more focused on just the mechanism of the cell death? Or are you also kind of looking at maybe what therapies may be even out there right now to stop it, even if you don't understand why it's being stopped?
Yeah, so I didn't really describe that too much, but I am really, so I think the first part of my research in my graduate experience was to focus on what cell death pathways are occurring and which one is the dominant one really in the neurons of our multiple sclerosis mouse models. And then when I figured out the cell death pathway, Parthanatos, that seems to have a huge role in MS, my next goal was to confirm not only that it occurs, but if we can block that cell death from occurring and if that had protective properties in the neurons, really. So that's where my research is focused. So we utilize genetic models. So these genetic models essentially have a deletion of a protein that is essential for the cell death pathway. So if we delete that protein, then the mice will not be able to carry out that cell death pathway. So that's where I studied a lot of my initial work, and then eventually moved on to a little more of an advanced topic related to pharmaceutical collaborations and such, where a pharmaceutical biotech company had developed with the Dawson lab at Johns Hopkins this Parthanatos cell death inhibitor. So this is something like an oral inhibitor that can be, given to mice, and that's where a lot of my research then moved to see if this is something that can potentially be used in patients if it works in mice.
So from your perspective, we hear a lot about many of the experiments are done with mice or rats, and then now we've got this kind of new technology with ergonoids or organoids and ISPCs and brains in a dish. Where, from your perspective, where are we in that transition? Is there still a role for both? Where do you, with what you're studying?
Yeah, so that's also a huge topic for research. So I think the mouse models and the cell culture dishes, while they are slowly becoming maybe less relevant as we have closer aligned preclinical ways to assess biology, really, they're still essential for the earlier phases of work. So like I said, I was trying to understand even what this cell death pathway is. And then I wanted to understand if it could, potentially be inhibited. And I think those early discovery stage research projects are really important to do in something like cell culture dishes or mice, where we can really harness the power of experimental manipulations so that we can get things just right to understand exactly how they're working. And then from there, I think they can, a lot of this research can be brought, like you said, into something like an organoid, or we can use advanced techniques like transcriptomic single nucleus sequencing analyses, things like that, on something like organoids or iPSCs that are derived straight from the patients before we progress into humans. So I think there's definitely a ladder of how we want to move on with the research.
I think, in today's environment, there's a lot of discussion around research and what universities are doing, and we probably don't want to go there. But what I do want to kind of probe on a little bit is really kind of the process of research, because first of all, researchers don't get funded large quantities of money right out of the start. You've got to prove concepts. You have to have that kind of basic information, which tends to be maybe a little smaller chunks of money. And I know the Karen Toffler Charitable Trust gave you a small grant, but it was kind of key in that understanding. Can you talk about, that importance of some of that initial seed money, but also, some of the complexities of really that research and funding and the challenges that while you're trying to learn?
Yeah, definitely. So I think, like I said, related to the experimental aspects early on, when you're trying to understand whether something even happens or it doesn't happen, that's where I was at earlier on. So 2022, really, and 2023, figuring out whether this is occurring. And that's why I applied for the grant. I was able to utilize a lot of these cell death protein markers that are expensive, to say the least. And a lot of the different ways that we're able to recapitulate the cell death pathway occurring in a dish. So that's where a lot of the funds went initially. And that was pivotal. That was a really pivotal moment for me, because I was able to do exactly what I wanted to do. And I figured out that this was a hit. This was a project that will actually carry me on for the next 3 years. And that's where I think a lot of that early seed funding, like the Toffler Scholar Foundation provided me with, it's really important, especially for researchers early on in the field that don't have, a lot of external funding. I think those smaller grants that allow them to explore something that provides the opportunity for the researcher to hit a topic that could be something that's really a hit. It could be something that's really interesting and potentially meaningful is essential. Because if we don't have that exploratory research at an early phase, then we'll kind of get stuck in the loop of recycling what we already know and doing research that we kind of already have seen. Maybe it works, maybe it doesn't really work, but it's the safest bet. And I think that's where a lot of the grants and such can kind of, it doesn't provide the most foundational research, I think. And that's why that early funding is so important, like what I've been given.
That's great. And I think you've had a publication or a couple of publications recently that kind of helped you with maybe further funding. Am I correct on that?
Yeah, you are. So I had, with these initial experiments, I was able to formulate a larger formal grant to the NIH and received an F31, which is predoctoral funding throughout a few years. And this came later on. Like I said, a lot of times people have external funding, especially as a professor, which I'm not at that stage yet. So I think having that initial project, those results, was really important for me to apply then as a later fourth year graduate student to the NIH and receive funding. And like you said, the publications are also essential. So I was able to publish or assist in publishing 2 smaller projects that are somewhat similar to my larger goal and larger project, which hopefully will be out in the next few months.
That's awesome. Yeah, you've talked a lot about, you learned this and then you went here and you learned that. And I mean, you've been everywhere from cancer to MS to Alzheimer's, and you've worked in some pretty amazing labs too. What, this interdisciplinary kind of collaboration or environment, how important is that too?
Yeah. So that's a crucial part of science. And I think anyone will tell you that, but to really delve into multiple fields at the same time, I think is not only exciting and provides you with, although it's tough, it provides you, and I'm learning every day with new knowledge and ways to look at your own research. So incorporating something like neuroimmunology or multiple sclerosis research, which is so heavily involved with the immune system, as well as the neuroscience, neurological, neurodevelopmental, neurodegenerative aspects of things, it's really powerful because, like I said, you're able to look at your research from 2 completely different fields, or maybe even more fields. And this holistic approach to research, I think, is also becoming slightly recapitulated in the medical field, where clinicians are trying to collaborate more with other specialties. So while you may have a cardiac issue, there could be a lot of issues also related with the GI tract. And these 2 fields, while they're completely different specialties in the clinic, they really need to collaborate because some drugs may interact with some systems and affect other systems. And you kind of understand how important that holistic approach to understanding the human body is. And I think that's super essential for research as we move forward, because you actually brought it up earlier. There's so many drugs out already, especially, related to cancer biology, that neuroscientists can utilize. And we're seeing this more and more as neuroscientists utilize, for instance, a diabetes drug that has always just been used for diabetes, specifically no relationship to studying neurons, is now really being looked at in psychiatric as well as neurodegenerative diseases. So that's kind of the idea of this interdisciplinary laboratory research driven venture.
Yeah, and unfortunately, a lot of patients don't, through their doctor, don't get exposed to the research side, don't get exposed to that interdisciplinary side of that. How do you think? I mean, because you've seen it in your own experience. I'm sure your discussions with your father, because he's talking to doctors too. But it sounds like he's a super curious person that's probably poking and prodding on the doctors to look here or look there, or what about this? Where do you think, just kind of looking into the future, into your future kind of career, do you think these two things will come closer together, medical and research? How do you think that's going to evolve?
Yeah, that's you touched on like one of the closest things I think to my heart and what I'm passionate about. I think seeing the disparities between how my dad is treated by a physician at a small private hospital in California compared to how my boss at Johns Hopkins, who's the MS research coordinator, seeing the differences between how patients are treated there compared to my dad is something that's on my mind a lot. So we often see these leaders in research and medical research. They are paving the way for new research and new therapies. So whether that means they know of biomarkers to assess for in patients, or whether they know of drugs that can be utilized from different fields to benefit their own patients that are, would never have been given these drugs, really comes down to whether the clinician is research oriented or not often. And that's not the clinician's fault at all. They're trained in a specialty. They go through a ton of practice in medical school. And then oftentimes these clinicians will stay in that field and that's their end goal. And of course they have to stay caught up to date and such. But where we see the difference, where I think I see a difference in how my dad's treated versus how patients under my boss are treated, is that if the clinician is research oriented and they're able to stay on top or paving the way of new research, they will treat the patients differently, and it will be more rigorous, it will be more up to date, and it will also be much more beneficial because treatments change so much over a time span. Over the past 3 years, over the past 6, 12 years, they have changed so much. And I'm witnessing just in my PhD how much they're changing in this 4 or 5 year time span. And I think that's really essential because new research is done every day. So much comes out every single day. A paper comes out and it completely changes how one biomarker is assessed. So I think I didn't really answer, but I think it's really important that this research and medical physician, clinician, scientist role really come closer and closer together. And I think that medical institutes like Stanford and Johns Hopkins are able to integrate that into their practice because they're teaching universities and teaching medical institutes. But outside of that, at the private practices and maybe the hospitals pretty removed from research universities or pharmaceutical spaces may lack that. And I think it's really important to figure out ways to integrate them.
Yeah, I do too. And I think it's, it kind of places a lot of the responsibility on the patient or the patient's family to try to, do enough research if they can to bring that up to their physicians. But that's really hard too, because reading the papers or even finding the papers, I've been through PubMed and it's not easy to navigate.
Yeah. And that's where also like scientific communication is so important for just researchers. Anyone can take part in it that is interested. I think scientific communication, being able to say what research means and potentially drive people towards looking at certain diseases in a different light and bringing what they are able to understand from these scientific communicators to their own lives so they're more informed and it's not so difficult for them. Because sifting through research papers if you're not in the field is extremely challenging.
Yeah. I may have this wrong, but I believe you're a pretty active mentor. You're in some mentor programs that are helping the folks behind you. Did I get that correct?
Yeah, there's a few different ones. That's what made me think about the scientific communication. So I've been a pretty active teacher, teaching assistant, tutor throughout the past few years, also an undergrad. And I think that's just a rewarding experience because it's fun to see students learn something and get better at it and then feel good about it and get excited rather than turn away from something that may be difficult, because I've definitely faced that and that's hard. So I think the teaching aspect of harder fields like neuroscience is really rewarding. I think teaching the general community outside of just the academic educational bubble is also really fun to do. And that's something I've really had the opportunity to take part in throughout Hopkins. One big thing that comes to mind is Project Bridge, which is just like the name. It basically bridges the general population, local communities with the scientists at Johns Hopkins. Project Bridge is at a lot of different universities, but at Johns Hopkins, I'm really into the scientific communication committee. So we take part in a lot of different ways to explain and showcase neuroscience and other fields as well to the population. So we have newsletters, we have teaching centers outside of farmers markets, we have science gong shows at breweries and at coffee shops. And then we also do teachings, things like Brain Week or something that recently happened, where we go somewhere in the local community and we pull up tents and we give out beanie babies that are neurons and we describe them. So that's always been really fun to connect with the community and feel like you're having some impact towards their understanding of difficult but exciting fields like neuroscience.
Yeah. Have you had one of those people yet come to you and say, you were the spark?
I have, but no one from the local communities or the younger kids, but I have had people say that I directly have taught or tutored. So that has mostly been undergraduate students.
Yeah. That's a wonderful feeling, I'm sure.
Right.
Yeah.
It's amazing.
So you're going to defend in about a month or so, you hope.
Yeah.
What comes next for you? And, what are you most hopeful for the future of your profession and your career specifically?
Yeah. So it's always fun to figure that out. And I think I know one thing for certain, if it wasn't evident, is that I do love the research, although it can be very challenging at times. It's always worth it in the end. So I'm going to stay in the research and discovery field of neuroscience. Of course, whether I end up at some junction between a biotherapeutic company for neuroscience research or end up at an academic institute, I'm still not exactly sure. I'm probably going to end up doing the typical pursuing a postdoctoral degree at a university after my PhD wraps up this year. And I'll probably go somewhere else outside of Johns Hopkins to get, more exposure to other places. And I'm not sure exactly what disease I'll be focusing on. I know it will be a neuro disease. Maybe it will stay MS. We will see. But it will definitely be something related to neurodegeneration and neuroinflammation and such. And I know that my main goal, like a lot of other scientists, is to contribute to or maybe have the privilege of really helping to develop therapies or a certain therapy that targets the disease and mitigates the pathology and the clinical symptoms in patients that they see in that disease.
I think we're all very grateful for you and people like you with that passion and that drive and that desire to stick it out and to do that, because we all benefit. We all benefit from that. So, Jackson, as we wrap up here, how can people get a hold of you? What's the best way to find out about your research, maybe fund you, maybe recruit you, whatever that may be?
Yeah. So I think my LinkedIn is a great place. I'm pretty active on LinkedIn. So it's just LinkedIn dash my name, Jackson Mace. And then I also have an email that I'm always on. So that's jackson.w.mace, M-A-C-E, @gmail.com. And then you can always look me up. I have pages on the Johns Hopkins websites and such that have emails and phone numbers.
No, I really appreciate your time today. I know you're busy. You're sitting in the lab. I disrupted your work, and
no, I needed it.
So I really appreciate it. I'm going to give you the last word as we totally wrap up here.
Yeah. No, I really appreciate having the opportunity to come here and utilize this platform, and I am beyond appreciative, and I will always, throughout my career, be appreciative of the Karen Toffler Foundation and the way that you guys have elevated my research career and how you elevate so many careers and give people the opportunity and the platform, like I said, to not only take part in research, but collaborate with other scholars and learn from other scholars. And this entire facilitated environment and community that this foundation has created is something that is going to be everlasting for me and a source of motivation and help. So I really appreciate that, and I'm happy to have been here and talked about my research and everything else.
Wonderful. We're going to have to wrap back by together here once you defend and you move on to your next phase. So again, thank you, and we will talk very soon.
Sounds good.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.